Good afternoon, everybody. I'm just happy that this time they did not give me a carcinoid diarrhea talk right after lunch. Been there, done that. Y'all probably know me. My name is Aman Chauhan. I'm a medical oncologist, and I recently moved to sunny Florida, University of Miami, to start a multidisciplinary neuroendocrine tumor program there, and also co-lead our theranostics program there. Let me see. Okay, these are some of my disclosures. So the topic of my talk is, what is the current landscape of clinical trials? And it's unfortunately one of those topics where you have to constantly update yourself, even as a NET specialist, because there are new trials coming on, old trials closing. So the landscape continues to change, which is a good thing. So we have to continue to, but why do we need clinical trials? Why is it needed? How do we improve our field? It is with the clinical trials. That's our only tool. I very well remember when I was a resident in New Orleans, and there were very limited treatment options. I remember our patients going to Europe to get PRRT. Or in fact, India had access to PRRT before US, right? So if we do not innovate, we'll be lagging behind everybody else. And in my career, in last 10 years itself, I've seen a big seismic change where we've not only catching up, but we are actually superseding Europe, and for that matter, other areas of the world in new drug development. And I'm here to kind of share throw some light on what's really happening. I won't really say these are the latest and the greatest, because some of them are my own studies, and I might be biased. No, I'm kidding. My studies are always latest, the greatest, right? I'm kidding. But it's just very difficult to talk about every single trial. There are like a lot of it. And even for seasoned medical oncologists, we don't know the finer detail of every single study. So the goal here is not to overwhelm you with what's happening, but the goal here is to show you there is the light end of tunnel, that we are doing a lot of really cool science that you have access to. So most of the studies would have this number called clinicaltrials.gov, clinical trials identifier. So you can take down that number, Google search it, and find more details about that trial. Where is it open? Is it open closer to you? Is that something you would be interested in, etc. But don't worry about knowing every single detail about that study. That's your oncologist's job. Or your oncologist needs to send you to somebody who does that trial, and then they would explain, is that the right trial for you, okay? All right, so as I mentioned before, why do we need to do trials? Because that's how we innovate. And what are clinical trials? These are the scientific tools, the methods by which we develop new treatments, new ways to diagnose cancers, new ways to prevent cancers. So this is just a scientific way to validate or disvalidate something, right? And what are different kind of trials? So any clinical trial that we develop, we try to have some good science behind it. We don't like to try a new drug or new intervention or a new scan into humans directly, right? So there is a lot of lab data, sometimes years, if not sometimes decades, that goes into understanding the science behind. And once we are ready and we believe, okay, this is effective in animals, this is safe in animals, only then we are brave enough to try first in human studies, okay? And those are called phase 1 studies, where our goal is to really prove safety of this intervention in humans, right? So these are smaller studies. These are not hundreds of patients. These are usually 10, 15, 30 patients. And the main goal here is to see what is the safe dose of the medication, right? Something which is unique to cancer trials is that these phase 1 studies are not placebo studies. So you do get real stuff, okay? So I want to assure you that, because many a times the phase 1 trial participants would be our patients who do not have standard of care therapies left. And we are really struggling for treatment options. And phase 1 trial gives us access to literally the cutting edge stuff, okay? Which might not be available in the commercially market for years, if not decades. So you get access to something really, for example, somebody who got PRRT here, you know, PRRT was approved in 2018 in the US, but people who got PRRT under trial, they got PRRT 10, 15, 20 years back, right? So early access is the advantage. The disadvantage is we don't know. We don't know if it will work. We don't know if it will be safe. That's why we do these studies. However, there are safety mechanisms in place. That's why phase 1, not anybody can do phase 1. Usually these are centers with phase 1 units, nursing staff, doctors trained to do these phase 1 studies because there's a lot more close monitoring and reporting, right? So we have to report anything serious to FDA right away. They give us like 12 hours, 24 hours, you know? So there's a lot more eyes on the patient. So, and there's a lot more closer monitoring, unlike standard treatments. A lot of phase 1 would involve a lot of blood testing every week, every 2 weeks, etc. So I tell my patients, actually, that's good. I feel comfortable because there are a lot more eyes, more than me, right? Somebody's taking care of you. You have somebody's phone number, research coordinator. Hey, I'm kind of having this issue. So I really like that aspect about phase 1. Phase 2 trials are a little bit more elaborate studies. Once in phase 1, we have proved the safety of a medication and what dose of medication is safe. Then we test it in a little bit more patient population to test, is it effective, and get early signals of efficacy. And the most definitive study is phase 3, where we have to treat some patients with the drug and other patients with the control. That could be either placebo. I know most of us I don't like placebo, but sometimes to get best science out, we have to have a placebo control trial. Other time we use best standard of care, okay? And this is how we really confirm whether our intervention makes a difference or not. Because you don't want to commercialize a product without really knowing whether it's helping. Because if it's not helping, it certainly is additional financial toxicity, side effect risk, etc. So we really want to make sure that our treatments are hitting one of the 2 goals, which are prolonging patients' life, second, improving quality of life. Okay, so those are the phase 3 studies and objectives, why we like to do those, okay? Look at all these clinical trial drug development and what has that done to our field. Because of these clinical trials, we now have not 1, not 2, but multiple treatment options. And it wasn't one-day effort. It took us years and decades. That's the landscape of drug development so far in neuroendocrine cancer. It's a slow process, but it is a robust process. The stuff that we have now, which is approved, we know it works for sure. It has went through the rigors of phase 1, 2, and 3, and we know this is good. It is not experimental, you know. A lot of patients tell me about experimental stuff. Hey, this or that thing, right? And I keep my mind open because I'm an investigator. But I tell them, do it as long as it's not affecting what we are doing, which is already proven. But at the same time, don't be misled because of these investigational stuff. It's not proven yet. For us to say that Lutathera works, or Zermelo works, or lanreotide works, we have already proven it. So I can say with conviction that this works, and this is the degree to which it works, okay? So moving forward, this is what we have so far, but we have to do a lot more. We have to catch up with our colleagues in breast oncology, colon cancers, leukemias, where they have even better treatments, multiple treatments, okay? So we are just scratching the surface, and it's the drug development which really lured me towards the neuroendocrine cancers because I felt that there was a big need for improvement there. And also, this is a very exciting aspect of oncology where you're able to bring the latest and greatest to your patients way earlier before it's commercialized. So you've heard a little bit about somatostatin analogs in Dr. Del Rivero's talk in functional nets. right? So we have great drugs, and I told you earlier, it's the backbone of neuroendocrine tumor treatment. But we have to constantly evolve as well. We have to give more options. When I was 1 year old, the short-acting octreotide, the one which we inject for rescue, the data on that came out. From there, it took us a couple of decades to get to the long-acting, Clarinet and Promid, but now we have that, which is great. Patients don't have to inject multiple times a day, right? But it's been a growing clamor in our community that can we have a pill version of the shot? And now we finally have a clinical trial which is testing the pill. It is not first in human study. There's already plenty of humans being treated with this pill for other endocrine diseases. So I'm glad that when I talk to my patient, that it's not, we already have tons of safety data. We know it works for that. Now we are trying to prove, does it work for carcinoid syndrome or not, okay? And this study is open at various centers, and we'll very soon have it open in Miami. We had it open in Kentucky. So please check it out, and this could be a reasonable option. Some of the things I like about the study is they cover for logistical costs like travel and stay, which is huge. Very few trials does that, okay? And we really lobbied for it because these out-of-pocket costs can really stack up, and we understand that. The other thing is a short trial. So after the screening is done, the active treatment part of the study is just 2 months. So it's a short and sweet trial. If the drug works, you get to continue the drug as an open label extension, and the company donates it to the patient. So that's a win-win for our patients. So it's a really wonderful way to get a little break from the injectables, okay? The next study in the same arena, testing a somatostatin analog, this hormone, is sort of led by my good friend, Dr. Singh from Canada. It's an industry-sponsored multi-center study, and it's testing somatostatin analog in a subcutaneous injectable form. So it's not once a month deeper injection, but it is the sub-Q injection, which is less painful, it's easy, and you can self-administer. I think that's the big thing, okay? So in the clinical trial, I think first couple of injections are done under supervision, but once patient's trained, you can do self-administration. And that's really, I think, the key takeaway point for that drug is that you can do it yourself. And it is done every 2 weeks. So there are these pros and cons. It's not once a month, every 2 weeks, but at least you can do it at your own comfort. So another important study, and this study is looking at tumor control. Unlike the paltusotine, the first study is carcinoid syndrome control. This one is actually looking for tumor control, okay? So that is going on, and that's the clinicaltrial.gov identifiers. You can check it out. It's a multi-center study. Again, you might have this access to this study closer to your center. And the other group of studies which are really popular in our field right now is our PRRT-based study. We had some great lectures from our theranostics faculty earlier today, so I'm glad I won't go too much into the concept of theranostics. You already understand why we do gallium or copper scan and how that translates into targeting these with lutetium-based PRRT treatments, right? So PRRT is already standard of care. It's already prime time, right? But is there role for improvement in the current data? Yes, so I'll be discussing some of those PRRT-based studies. First of all, we would like to now understand, is PRRT better than chemotherapy, CAPE-TEM? There were some questions in the morning, right? Between CAPE-TEM, etc. So now we have two options which are standard of care for pancreatic neuroendocrine tumor, PRRT and CAPE-TEM. So a lot of my patients ask me, hey doc, which one should go first, right? So we currently use our clinical judgment, we understand the burden of disease, side effect profile, et cetera, and can try to. But this study would give us objective scientific data, which one is better, and which one turns out to be better, maybe eventually ends up being the one which we go first, okay? So this is a good effort by our colleagues. Dr. Soares is co-leading this important study. Also, the NETR-1 study, which led to FDA approval of Lutathera, only treated mid-gut, that means small bowel patients. They did not include pancreatic patients, although the FDA approval is for the pancreas and midgut. So this study will also give us prospective data for pancreas NET patients. Okay. The other important study which is happening right now in the country is a multi-center study led by my good friend Dr. Hope from UCSF. This study is investigating the role of lutetium-177 dotatate in lung nets. See, the FDA approved it for GAP nets, or lung patients, which are about 30% of our patient population. They're left out. So sometimes insurance would give me grief that, oh no, we are not covering it, right? So studies like these are very important to generate prospective data so that we can support, hopefully if this is a positive study, that this drug also work for somatostatin receptor positive lung nets. So it's a very important study evaluating PRRT in lungs and comparing it to our standard of care, which is everolimus. Next study I'd like to talk about is a small lag, is an industry study called COMPOSE. It's a very, very important study. Why? Because it's investigating PRRT in an area which is critical unmet need. That's our high-grade, well-differentiated neuroendocrine tumor patients. So higher grade 2 and grade 3 well-differentiated neuroendocrine tumor patients. Grade 3 well-differentiated NET is a very new entity. Before 2017, we didn't even know that this existed. Previous WHO classification lumped all grade 3 cancers as one, poorly differentiated, which was inaccurate. Then we realized that not all grade 3s are same. So there are some poorly differentiated neuroendocrine cancers, and there's some well-differentiated neuroendocrine cancers. And they behave differently, different biology, and treatment should be different. So in 2017, WHO finally recognized this grade 3 well-differentiated NET. But since this is such a new entity, we don't really have a lot of treatments for it. So studies like COMPOSE really helps us bridge that gap. So that is an important study. We are going to hopefully open it in Miami soon as well, but it's already open at various centers in the US. So another important study. So we talked a lot about the PRRT, the lutetium-based PRRT. There is a newer type of PRRT also creating a lot of buzz these days, and that's called alpha PRRT. So lutetium-based PRRT, the lutetium-177 dotatate, it causes the tumor damage because it releases what's called beta radiation. Okay? The other radionuclide like lead-212 or actinium, they release alpha ionizing radiation. Okay? So they're different type of ionizing radiation. So some people believe that alpha might be better. Of course, that is yet to be proven. But theoretically, what alpha radiation does is it is shorter in the duration, so it doesn't penetrate deep. So theoretically, your normal tissue doesn't get exposed. And it is higher energy, so it causes double-stranded DNA breaks as compared to more single-stranded DNA nicks with beta. Again, all theoretical, and all of this needs to be proven in humans. But I want to share this data. So the phase 1 study evaluating lead-212 dotatate from Dr. Del Poison's group, they treated some patients with alpha PRRT, great results, and you can see one of the pictures there, the tumors sort of disappeared, okay? But these are anecdotes, they have to be proven in larger studies, okay? However, very promising newer agent, and is currently being investigated in larger studies, okay? Similar alpha particle studies are also, I'll talk about, being investigated by another industry using actinium-based drugs. So these PRRT studies were for frontline PRRT. That means patients who have never gotten PRRT before. Now we have some trials for patients to cater to the patients who already have PRRT, because that is also a big patient population, right? Because many of you might have already gotten standard of care PRRT, okay? So there are some trials looking into those patient populations. So one of those is, do you know that guy? The smart looking one on the right side? I'm kidding. So I had this opportunity to co-lead this international study with my good friend, Dr. Singh from Canada. It's a joint US-Canadian venture. It's a government, so you and me are paying for this, our tax dollars, okay? So we are hoping to generate data in support of that PRRT can be done at progression after initial PRRT. So right now, the PRRT is approved only for 4 doses in the United States. So if I have a patient who had good run with PRRT, stable disease for 3 years, now after 3 years, the tumor starts growing again. His automatic question would be, doc, this worked so well for me, 4 doses and I was fine for so many years, let's try it again, right? And we can, because there is a lot of data from Europe which suggests that it can be safely done again. And this data also suggests that it is effective again. But those are all retrospective, single-center data. So insurance can come back and say, it's not approved, we are not paying for it. So studies like these are very important to prove whether if we can retreat with PRRT safely, and if this is positive study, then hopefully opens the gateways for access to other patients for standard of care, okay? So this is a study which is about to be launched next month or two, and we named it Net Retreat. The other study I want to mention in the repeat PRRT setting or post-PRRT setting is this alpha emitter PRRT study. It's a phase 3 study, industry-sponsored study, looking at actinium in this patient population. So patients who have already had standard of care lutetium dotatate at progression can consider alpha PRRT if that's something they're interested in. Next up, so those were some of the key PRRT-based studies. What's happening in non-PRRT arena? So Dr. Suarez again, and her surgery colleague, Dr. Ahmed, they are testing role of CAPE-10 in early stage neuroendocrine cancer tumor of pancreas. So you've probably heard about adjuvant chemotherapy. What does that mean? So if we have caught a tumor early in the stage, the best treatment is surgery, just resect it and we cure the patient. But we do know that some patients, the cancer comes back and relapses. Why does that happen? Theoretically, that happens because there's some microscopic disease left behind which no surgeon can see, no scans can pick. In those scenarios, we often give chemotherapy for for limited time period after surgery to kill all the potential microscopic disease in hopes of prolonging the disease-free or recurrence-free survival. That's a very common thing in a lot of cancers, colon cancers, breast cancers, where patient gets the breast surgery and then gets some chemo or radiation after that. In neuroendocrine tumor, we just don't know if that strategy is really good or not. So we are testing adjuvant chemo with pancreas and not every pancreas. So there is a risk criteria, high risk patients, so there's size criteria, grading, etc. So if we believe that is a high risk patient for relapse, then we are considering this clinical trial where we give 4 months of chemotherapy so that we can improve the relapse-free survival. And this is a study ongoing right now. The other study, which I'm very excited about, is led by Dr. Jennifer Chan from Dana-Farber, and is testing a pill-based targeted therapy called cabozantinib. It's a VEGF inhibitor. What is VEGF? It's a vascular endothelial growth factor. So tumors grow in our body for various reasons. There's numerous pathways, but one of the pathways is that it is able to develop new blood vessels to feed itself better. And this particular drug tries to clamp down on that. It tries to slow down the new blood vessel formation in the tumors, okay? Among various other things that it does. So this study is evaluating whether this pill called cabozantinib will slow the tumors down, and it is building on some successful phase 2 cabozantinib study where we did see that it's very effective in a lot of neuroendocrine tumors, especially pancreatic neuroendocrine tumors. So it's a very exciting study. Of course, since this is a phase 3 study, this is a final step. If this study is positive, this leads to another approved drug. So this is a really important study. If it's negative, of course, we go back to the drawing board. So very important study, but because of that registrational nature of the study, there has to be a controlled placebo arm. Otherwise, we would not know whether this drug works or not. But the good thing here is, if, first of all, only 1/3 of the patient gets into control arm, 2/3 patients get the real stuff. But those 1/3 who do get into placebo, at progression, they can get the real stuff. Okay, so at least there is a crossover allowed, which is a good thing. So I don't feel that our patients are kind of getting, any issues. All right, so next up, that's our good friend, Dr. Del Rivero. This is the study. I think there's a question regarding this study. I was reading the patient question. It's like, what is that pill that Dr. Del Rivero is mentioning about? So this study is looking into chemotherapy plus a targeted therapy called PARP inhibitor. And the reason why they have decided to combine these two is because the chemo causes DNA damage, but the cancer DNA has capability or power to repair itself. So this PARP inhibitor is supposed to impede that process. It delays that DNA repair process. So it really works hand in hand. Okay? So Dr. Del Rivero's study is looking into this temozolomide versus temozolomide plus olaparib, which is a PARP inhibitor, in this very rare neuroendocrine tumor, which is also another critical unmet need. We don't really have good treatments for this cancer when it's metastatic and progressive. So I'm glad they're doing this, and if it's positive, I wish them all the luck. We have one more treatment options for our patients. And I would like to conclude the last segment of studies with some of the studies that I'm personally involved in. I'd like to give a little shout out because, of course, they're better than all studies. I'm kidding. But theranostics, you've kind of heard that term, right? So this field of oncology is catching up big time, right? So what does theranostics mean? If we know that there is a target in the cancer, right, we can develop a drug to target that, and that drug kills the cancer with the help of radiation. So it's a precision way to give the radiation. For example, you see here, there is this cancer cell, and there is this target. Let's take an example of neuroendocrine tumors. What would this target be? Somatostatin receptor. So if we have a drug that likes to go and bind to somatostatin receptor, and we have that drug, right? All of us are getting this in injection, somatostatin analog. So if we have an analog that likes to go to the receptor, what if we bind this analog to a radioactive heavy metal? And that is lutetium-177. So we inject this drug, it goes and binds to the receptor, and it drags along this lutetium-177 with it. So it really takes this radioactive compound to the cell and kills it. That way we don't affect the normal healthy cell. It's the precision way or tailored way to give effective treatment right at the tumor. What if we can do this for any cancer like breast cancer, HER2, colon cancer, fibroblast activity? As long as we have a target, we can use the same concept to develop new innovative tailored drugs for cancer. So this field is investigating it. Look at these. These are not NET patients. These are breast cancer, pancreatic cancer, colon cancer patients, different target, same concept. So I personally believe that this field of theranostics is going to grow, and we are just scratching the surface. But I'll come back to NETs. That's my area of interest. So PRRT is now mainstream, right? Should we stop the further drug development here? No, because we now have to look at what are the next generation of problems. Can we improve on our current PRRT? Maybe we can do that by alpha. Maybe we can do by some combinations, right? Maybe we add chemotherapy to it. So there are different ways we have to think, because the current PRRT works, but doesn't work as well as we want, right? The overall response based on the FDA label for Lutathera is 14%. That means it only shrinks it up to 14%. So if I treat 100 patients, only 14 might show shrinkage. Can we make it 50%? That's what we are shooting for, okay? The other issues that we now have is if somebody has progressed to PRRT, how to best treat them, what's the radiation resistance, why it happens, how to overcome it, how to stack treatments, how to sequence treatments. So those are all next generation of problems that we have to tackle. So one of the studies that I first got involved in my early career is a drug, testing a drug called triapine. It's a pill. It's a radiation sensitizer. And we added this pill to our standard of care drug, Lutathera. So see, Lutathera is given IV, 4 doses, easy peasy. But the pill complements that. And we give this pill for 2 weeks during each Lutathera cycle. And the way it works is the Lutathera causes DNA damage of neuroendocrine tumor cells, and triapine impairs the repair process. Okay? So we started this process in our lab in 2018. We were able to enroll our first patient in 2020 because the lab data looked really good. And I'm happy to say that as of January in GI ASCO, we presented the pharmacokinetic data. So the phase 1 is officially done. And more importantly, we are making it to a randomized phase 2, which will be launching next week. So you're really hearing it from the first one to know, right? So this is really gratifying to know. I mean, you can see it's years of effort, right? And I'm building on people who have worked, for decades before that. So triapine was first developed at Yale and has been in development for 20 years. And after that, we started working it on NETs. So it really takes a village. It takes a lot of time. So let's see if triapine really adds a punch. Does it make Lutathera better or not? So this randomized study would give us some answers over the next 2 years, okay? And this study will be open at various sites in the US. We expect 20 plus sites. So very, very excited about it. Not only we are trying to improve the therapies, we are trying to also improve the way we detect these cancers. We all kind of have to get scans, and with chronic diseases, sometimes it gets like too tiring to get scans every 3 months, every 4 months. Can we build some good blood tests to monitor? As of now, we don't have a good blood test that can replace a scan in NETs. In 2021, we stumbled upon a blood-based biomarker called HPG-80, and we tested it in about 92 neuroendocrine cancer patients, and we found that it is elevated in neuroendocrine cancer patients. We presented that data. Now it's published. You can actually Google it in cancers. But the next best thing is to validate it in a clinical trial. So we are now validating it in a couple of randomized studies, and every time patient will get scanned on our studies, we'll also draw blood for HPG-80. And if we can prove that the progression, remission, disease status correlates with the blood level, that would be wonderful. And we might have a tool to help monitor disease with the easy to do blood test. And lastly, another study that we are currently doing is a multi-center study. Is using a brand new drug called piposertib. When I first started working with this drug, it didn't even have its name. It used to be called M3814. And the drug was shipped to me from Germany, from German Merck called KGA. And the way this drug works, it's a DNA PK inhibitor. Again, it is a very specific enzyme. Anytime our cells take a DNA damage, be it from sun exposure, radiation, or anything, our body repairs itself. So it upregulates the DNA PK enzyme, which helps in the repair process. So this drug is designed to cut off that process. It's an inhibitor of that particular enzyme, right? So we tested it in our mouse models for pancreas neuroendocrine tumor, and as you can see here, when we added piposertib to the radiation, the tumor growth really got stunned. This drug by itself doesn't do anything because you have to do the DNA damage, right? And then it helps prevent the repair process. So we are very excited about that study, and that study is ongoing. And lastly, we discussed about net retreat. So we are excited about that study being launched in the next couple of months as well. Dr. Del Rio asked me to give a couple of minute talks about high-grade. High-grade neuroendocrine cancer is completely off topic, and it really deserves its own 1-hour lecture. But one of our speakers who was supposed to come here and give the talk cannot. So I'll just give like 2 minutes on high grades, okay? Because we have some patients who have high-grade neuroendocrine cancer patients. So when I see a patient in my clinic, I tell them that neuroendocrine cancer is very unique. It's not one disease. It is a spectrum of disease, okay? And what do I mean? So based on the pathology, based on how it looks under the microscope, it varies from all the way from low grade, otherwise called as, in past we used to call them carcinoids. Now we prefer to call well-differentiated grade 1 NETs, to intermediate grade, well-differentiated grade 2 NETs, to high grade, which is either well-differentiated grade 3 or poorly differentiated neuroendocrine carcinomas, okay? In lung world, we still call them typical lung nodes, atypical carcinoid, and poorly differentiated carcinoma. But this is the whole spectrum of disease from less aggressive to more aggressive. So the first thing that we have to understand when we deal with neuroendocrine cancer, really know what we are dealing with. And it's very common for me to see when patients use these terms very loosely. Grade 1 well-differentiated neuroendocrine tumor labeled neuroendocrine carcinoma, which is a very different cancer. So it's really, really important to know which bucket are we getting into. Because the way I treat grade 1 well-differentiated neuroendocrine tumor is completely different than poorly differentiated neuroendocrine carcinoma. So first important distinction is, do you have NET, neuroendocrine tumor, or NEC, neuroendocrine carcinoma? T versus C makes all the difference. We will do gallium or copper scan here. We'll do FDG PET here. We will do treatments like PRRT, surgeries, everolimus, vasandanol. We will do platinum-based chemotherapy here. It's completely different, okay? And within the NET, what makes it challenging is different gradation, right? And we treat all of these differently. So it's really, really important. So high-grade neuroendocrine cancer, without going too much detail into it, it's a different cancer, more aggressive. It looks very different under the microscope. It harbors different genetic mutations, and it's treated very differently. So it's really important to understand what we're dealing with. And one of my areas of interest is also high-grade neuroendocrine cancers, because during my training in Kentucky, especially catering to rural Appalachia, this is a big, big problem there. Lung neuroendocrine cancer, small cell lung cancer, you've heard, is one of the most aggressive cancer that we know. 5-year survival of small cell lung cancer is still under 5%. And that's how it's been for past 5 decades. So it's a recalcitrant cancer, and we really need to do a whole lot in this area. So we are constantly working on developing new therapies. One of the top therapies that we are working on is based on oncolytic virus and immunotherapy for high grades. And I'm hopeful that we will launch a study sometime this year. With that, that's my next new project, my little baby. And that's keeping me plenty busy. And I'd like to thank you for your attention and this opportunity to kind of be here with you guys, and happy to take any questions later. Thank you. And I did it under time.